Chromosomal duplications. Gene duplications or the chromosomal duplications is a phenomena by which new genetic material is produced. As a result of gene duplication or chromosomal duplication, a particular fragment of the chromosome or particular sequence of the DNA is replicated again and again. Such a phenomena is called as gene duplication or chromosomal duplication. In this diagram, we can see that what is chromosomal duplication. If we do the comparison between two chromosomes, we can see that this part of the chromosome has been duplicated. Here is a single, but this part is duplicated here once again. So chromosomal duplication is the fragments of the chromosome, they produce their copies, or a specific fragment of the chromosome multiplies and there produces the duplication. Causes of chromosomal duplication. There are several causes of chromosomal duplication. One of the main cause is replication of the DNA. During DNA replication, the DNA polymerase slippage and it replicates the copies of a specific fragment of the DNA. But common cause of DNA duplication is ectopic homologous recombination. Ectopic homologous recombination is a phenomena that is usually between two misaligned chromosomes. We have seen that during the process of meiosis, homologous chromosomes they align with each other. But in this type of the recombination, they misalign with each other and there is exchange of the genetic material from one chromosome to another chromosome. Ectopic recombination is a phenomena which occurs due to the misalignment of the chromosomes. There are some other causes of DNA duplication or chromosomal duplication. Those phenomena are called as retrotranspositions, euploidy, aneuploidy. They are also responsible for the DNA duplications. Replication slippage is another phenomena that causes DNA duplication or chromosomal duplication. Replication phenomena is that the DNA polymerase slippage it start to produce copies of a specific fragment of the DNA. Here we can see the duplication process, how it occurs in the chromosomes. This is the structure of a chromosome where we can see the fragments D and E. While in this chromosome, this fragment has been copied. This is the first copy of this fragment and this is the second copy of the same fragment. In case of if there are additional repeats, the first copy, second copy, third copy and fourth copy. So this phenomena, when a particular fragment of the chromosome has been copied and it is placed side by side or it can move to another location, this phenomena is called as duplications of the chromosomes. Duplications of the chromosomes can be of two types. First one is called as dispersed duplications and second one is called as tandem duplications. Dispersed duplications can be on different chromosomes or on different locations of a single chromosome, while in case of tandem duplication, particular fragment of the chromosome is copied and it is placed side by side in the form of tandems or in the form of repeats. Tandem duplication becomes pseudogenes. Sometimes the tandem duplication is of a particular gene, and if there is a mutation in that gene, or that gene adopts a particular function in the chromosome. But most of the time, the duplicated regions or the duplicated genes, they are converted into pseudogenes. Pseudogenes are those genes which do not produce messenger RNA, or they do not have any function, but they are present on the genome.